Hello, my name is Vanessa Esparza, and for my special topic assignment, I decided to focus on the Hox gene, Tiktaalik bridging the evolutionary gap of fins to limbs. I wanted to start this presentation off like a story because evolution is really the story of progression of how organisms started to where they are today. So, once upon an evolution, there was a man named Neil Shubin. Now, today, Neil is a paleontologist and evolutionary biologist at the University of Chicago. His goals include studying how new features arise in the lineages of animals. However, he is most famous for his discovery of Tiktaalik rosea, the key to bridging the evolutionary gap of fins and limbs. But before we get too ahead of ourselves in this story, we're going to rewind a little bit and take it back to 1986. The fish to tetrapod story wasn't complete. The image on the right-hand side is actually an image that a young Neil saw in one of his lectures. He saw this image and was a little bit puzzled. He kind of asked himself the question, how did a fish with fins develop into a tetrapod with limbs? There had to be something in between. And so this kind of sparked Neil's journey into discovering what that bridging organism could look like. But before we get started, let's discuss the differences between a fish and a tetrapod. A fish, typically a cold-blooded aquatic vertebrate, which has gills, fins, and is typically an elongated body covered in scales. They have a conical head with eyes on either side, and their head is connected to their shoulders by a series of bones, and of course, they have fins. Focusing on the fish a little bit, the conical head with eyes on either side. They can't move their head independently from their body, and that is going to be a key role as we further on in this story. Now focusing on tetrapods, a four-footed organism, a part of the vertebrate group that is a bit higher than fishes. They have a flat, wide head with dorsal eyes, have a neck and head that can move independently, and of course, limbs. Focusing back on that picture that Neil saw in his lecture, he said there must be an organism in between, something that can bridge the gap between a fish and a tetrapod. Perhaps a fish that could move its head independently, with a flat head and dorsal eyes, but with fins, something in between, a hybrid between a fish and a tetrapod. But what is it exactly that Neil was looking for? Fossils. We've discussed in class a few times how fossils are definitely a bridge to connecting organisms in the phylogenetic tree. We need rocks of the right age, the right type, and rocks that are exposed to the surface. Having these three elements will lead us to the actual fossil that we need to bridge the gap between fish and tetrapod. And so rocks of the right age include rocks that are 390 to 365 million years old. We're going to focus on the Devonian era. Rocks of the right type. Sedimentary rocks are rocks that hold many fossilized organisms, so sedimentary rocks are definitely the rocks of the right type. Then rocks that are exposed to the surface, naked bedrock. Of course, we can't go digging for fossils that are thousands of feet below the surface. They're just not accessible. So we need something that is accessible for us to go out and explore in. I say us as if we were actually there with Neil, but watching these videos, it kind of made you feel like you were there. So now that we know we need rocks of the right age, rocks of the right type, and rocks that are exposed to the surface, where exactly do we look for these kinds of fossils? Going back, we're bridging that gap between fish and tetrapod, sea and land. Therefore, ancient seaways and ancient highlands, somewhere in between that transitional period. So delta systems are a great place to look. Neil identified 3 places in North America that would have these three elements that we were looking for in fossils: rocks of the right age, rocks of the right type, and rocks that are exposed to the surface. And these three places included East Greenland, the Catskills in eastern Pennsylvania, and the Arctic Islands. However, other paleontologists had explored the areas of East Greenland as well as the Catskills in Pennsylvania, which led to somewhere that was a little bit less accessible but still accessible nonetheless, and that was the Arctic Islands. Ellesmere Island, Canada, 700 miles above the Arctic Circle, was the perfect place that had rocks of the right age, rocks of the right type, and rocks that were exposed to the surface. The expedition to find the organism to bridge the gap between fish and tetrapods began in 1999 and ended in 2004. Above are some pictures of Neil and his colleagues looking for said organism, looking for any kind of fossil that had a flat head with dorsal eyes, a head that can move independently, and fins, a hybrid between fish and tetrapod. And in 2004, that's exactly what they found. They found a fossil of a flat-headed fish with dorsal eyes, a head and neck that can move independently, and fins with limb-like characteristics. So introducing the main character of this story, Tiktaalik, a flat white head and dorsal-eyed fish. It had a neck and a head that can move independently. It had both lungs and gills, and fins with limb-like components such as a shoulder, elbows, a forearm, and a wrist. The perfect hybrid between a fish and a tetrapod. So now that we have this fossil, now that we have Tiktaalik, what exactly do we do with it? We have our physical comparison, but we need something a little bit more, something that can genetically solidify the gap or the bridge between fish and tetrapod, fin and limbs. So it's time to bridge the gap, Tiktaalik. Tiktaalik allows us to compare limbs of different living organisms. Comparing Tiktaalik to living organisms allows us to solidify the bridge between the evolutionary development of fins and limbs. Having a physical comparison is only scratching the surface, and it is vital to understand how fins developed into limbs. On the bottom right-hand corner, we have an image of a phylogenetic tree. You can see at the very bottom of our phylogenetic tree, we have a fish with fins, and at the very top, we have our tetrapod with limbs. And Tiktaalik has a special place within the phylogenetic tree. It sits right in the middle, right where Neil knew there had to be something that bridged that gap. So again, we have that picture that inspired Neil to go on this expedition, and now Tiktaalik has a place right in the middle. Bridging the gap comparison, the comparative process. This consisted of doing a CT scan of Tiktaalik's skull. Comparing the CT scan of Tiktaalik to a living organism, such as an alligator gar, which is the picture at the very bottom of the skull, showed that Tiktaalik's skull has cranial kinesis. This means that the bones within Tiktaalik's skull can move independently, which is important to, again, bridging that gap between a fish that can't move its head independently to an organism that can, but this time has limbs. The discovery of Tiktaalik was only the beginning. Dissecting this fossil and comparing it to living organisms will then allow us to see the developmental process of fins to limbs. As I stated previously, it's great that we have a physical comparison. However, to solidify that gap, we do need some genetic evidence or some genetic comparison. The driving developments. The picture to the left is a picture of a fish, and to the right is a human arm. We can see the visible differences between these two organisms. Obviously, fish don't have fingers, humans have fingers, but beneath the surface, the way they develop is very similar. So driving developments. A set of interactions among signaling centers, genes, and proteins being turned on and off are the developmental driving forces. The picture in the image is comparing fish, human, and a chicken. We can see the developmental process is very similar. However, there are genes that are being turned on and turned off to maybe develop a hand instead of fins. To continue bridging the gap, we have the Hox genes. Hox genes are a family of regulatory genes that encode transcription factors that are essential during embryonic development. Hox gene knockout. If one of the Hox genes is eliminated, hence knocked out, then it can affect the developmental process of a limb. For example, the image on the bottom right-hand corner is a wild-type mouse arm. The first image has 3 specific sections. We have that top part of the arm, the middle section of the arm, and the hand itself. If one of the Hox genes were eliminated, say the Hox13 gene, then there would be no hand that developed. It would just be an arm without a hand. On the other hand, if we eliminate the Hox11 gene, we would only be left with that first segment and a hand. That middle segment would be eliminated. So what do these genes have to do with tiktaalik, fins, and limbs? They are responsible for the specification of the different components of our appendages. They are a key player to understanding the origin of digits from fins of fish. There are 2 phases in the Hox gene. We have our early Hox phase versus our late Hox phase. Early Hox phase. Different genes of Hox system expressed within one another. In the video, Neil explained this as Russian nesting dolls. When you open one, there's another one inside. So similar with the Hox system expression, there are genes within genes being expressed. So the early Hox phase acts early on in the limb development and is involved in the specification of the first 2 segments of the appendage. The image on the left, which I drew myself. The first 2 segments are circled, and those are what is being developed in the early Hox phase. The late Hox phase involves expression across the entire distal domain of what will become the digits and wrist of a limb. So again, going back to that image on the left, what's circled at the very bottom, the hands, the digits, the phalanges, I guess you can say, will be developed due to the late Hox phase. Late phase expression already exists in fish fins and perhaps is coding for something else. Now that we know that the late Hox phase gene is present in fish but might be coding for something else, means that we can take it a step further in the comparative genetic process. Patterns of activity of Hox genes in limbs and fins. Comparing tiktaalik to a paddlefish. The paddlefish was a good initial candidate for comparison because they produce a lot of embryos, which meant a lot of experimenting with them. And they also sat in a good position in the phylogenetic tree, similar to where tiktaalik sat. So going back a couple slides to our phylogenetic tree, we have tiktaalik right in the middle. Our paddlefish probably sat somewhere above tiktaalik, putting it in a good position as a candidate for comparison. Results show that the paddlefish had both early and late phases of the Hox gene, which meant that there was that early expression of the first 2 segments of the arm as well as the last segment. But again, it wasn't developing digits or a wrist, probably developing something else. Early Hox expression is on the telometric phase of the chromosome, while the late Hox phase expression is on the centromeric side of the chromosome. I guess we can say that these genes are linked on the same chromosome. Ha, get it? Okay, anyways. There was a problem that did arise, however, when taking the late phase enhancers from a fugo fish and putting them in a developing mouse embryo. there was no success in the driving late phase of expression of limbs, meaning when the enhancer was put into a mouse, the specific late phase enhancer, those digits and wrists did not develop, meaning maybe the gene is turned off or maybe it's expressing something else. So again, this means late phase expression is unique to the development of digits and unique to the tetrapod. Fish do have this gene, it's just not functional in the same way. Finding the right animals for comparison. Having the right animal could be a key to discussing or discovering the genetic connection between Tiktaalik and fish and Tiktaalik and a tetrapod. And so some characteristics or some candidates for comparison were humans to chickens, chickens to fish, and fish to humans. The results show that enhancers were not present in the fish fins. Therefore, it shows that we need a new animal for comparison. The picture on the left corner has our 4 comparative organisms. We have a human, chicken, a zebrafish, and a pufferfish. As you can see, there is activity of those late enhancers in humans and in chickens. However, there's no activity whatsoever in the zebrafish or the pufferfish. And this could be due to genome duplication. For example, humans have 4 Hox clusters, while living fish might have 8 or even 16. We needed a fish or an organism that didn't have that genome duplication, something that was a little bit more similar to a human or a chicken. And that brought us to the GAR as a comparison. The GAR has allowed us to properly compare these enhancers across different organisms and has even brought to light the enhancers in organisms that were not originally showing any evidence. So going back to that picture, we see there's a lot of activity in the human, a lot of activity in the chicken, but none in the zebrafish or the pufferfish. However, when we introduce the GAR, you can then see that activity being brought to light in the zebrafish and the pufferfish. Now, the question is the chromatin accessible in the right stage of development? Neil said him and his colleagues use a new technique, the ATAC sequence, and this shows the accessibility of the chromatin, specifically looking at the CN565 gene, which is in the picture, the one that is showing a lot of activity in the human, in the chicken, but none in our zebrafish or pufferfish until the GAR is introduced. We know that the late Hox phase, which is on the centromeric side of the chromosome, are the driving forces of digit expression. So the question is, do fish have the genetic apparatus that drives Hox activity, which drives the formation of digits? Well, first, yes. Yes, early enhancers are present in fish, so it's those first 2 segments of the arm. The picture on the bottom left in green shows those 2 segments of development, which are on the telometric side of the chromosome. And yes, the late enhancers are also present in fish. Going back to that picture on the left in yellow, the digits that are being developed. However, they report similar in development, but not necessarily in physical comparison, which is why we needed that physical comparison of fish to Tiktaalik, from Tiktaalik to the tetrapod. We see that physical comparison, but now we need a genetic comparison. So what is the big idea? What is the big idea that is taking us from a fish to an organism such as Tiktaalik to a tetrapod? We can take leverage from multiple kinds of data to understand our evolutionary history. If we go back to the image that started it all, we now can complete the story of fish to tetrapod. We have Tiktaalik that bridges that gap, and it's important because now we have a better understanding of how organisms have evolved over time. Three things that I found interesting within this specific topic were how genes can be turned on and turned off, or how there could be different phases of the same gene. So, for example, the Hox gene, there's an early phase and a late phase, and when one of those is eliminated, the development of how limbs look obviously are different. Kind of like a mutation. Obviously, I think this might be a little bit different than a mutation if something is eliminated, but it's just so crazy how a small gene can determine whether or not you have an arm with a hand or a hand with no arm. I also found it really crazy how the GAR was able to bring to light the enhancers in the pufferfish and the zebrafish when we initially just compared human to chicken to zebrafish to pufferfish. But with the introduction of the GAR, we were actually able to see those enhancers. It was honestly so crazy seeing how not only was Tiktaalik the key to bridging the gap between fish and tetrapod, but the GAR was also that key player to bridging the gap of finding these enhancers and comparing them between different organisms. Another takeaway for me was just, I guess, developing the idea that there has to be something in between a fish and a tetrapod. I remember even being a kid, I had these DK Witness books. I had a pirate one and an ancient Egyptian one, and I was so convinced that I was going to be able to find fossils and mummies in my backyard. And so it's actually really crazy how Neil was inspired by an image that he saw in his lecture and took it upon himself to go on this expedition and find the correct components, finding rocks of the right age, rocks of the right type, and rocks that are exposed to the surface, and actually going out into the field and looking for something that might not even exist, but taking that chance to see if it actually does. That was honestly so captivating and something that I took away and wish that maybe I could do one day. Maybe I could find a fossil in my backyard or something, but just the whole process of the expedition was something that was so captivating. And then actually going through the process of comparing genetically if this organism even genetically fit was really captivating. When looking at the developmental processes of different organisms, and when we compared the developmental process as far as limbs go from a fish to a human and a chicken, it kind of brought me the idea, and I know this might be a bit off topic, but how maybe some pro-life protesters would use images of a chicken embryo, not realizing that it is a chicken embryo and saying it's a human embryo. It kind of just shows that the way organisms develop across the board is so similar, and it really only takes either specific signaling centers, genes, or proteins to be turned on and turned off to determine whether or not you'll have fins or an arm or a chicken leg or something completely random. It's just so crazy how something so small can determine something so big. And that concludes my presentation on the Hox gene, Tiktaalik bridging the evolutionary gap of fins to limbs. I hope I did this presentation justice. There was a lot to take in and a lot of big words that I was introduced to, but I think I got the concept of the idea and bridging those gaps genetically, physically, and in the phylogenetic tree and in the genomes of different organisms. And so it was very interesting to watch this video, and honestly, like, Neil Shubin, he's a cool guy. I genuinely admire his work and his enthusiasm and his idea of just, well, what if there is something in between and what can I do about it? And sure enough, there was something in between, and taking that risk to go on an expedition is something that's honestly really inspiring. And so that concludes my presentation. I am Vanessa Esparza, and thank you for watching.